Gericke Cook's a mathematical statistician and risk analyst with the Transboundary Disease Analytics Unit at the USDA APHIS Veterinary Services Center for Epidemiology and Animal Health in Fort Collins, Colorado. Her work with the USDA has focused on models and risk assessments on the entry and spread of invasive agricultural pests and diseases so that surveillance efforts are appropriately targeted. She has worked with both plant health and animal health sectors, and has particular interest in understanding how humans introduce and spread invasive organisms. Human introduction and movement of gene drive containing organisms is an analogous problem during field trial stages of gene drive technology development, and Gericke's work should speak to gene drive technology developers. Gericke, thanks for your willingness to share your work with us today. Really appreciate it. And with that, I'm going to turn the floor over to you.
All right, thank you very much. That was a great introduction. Thank you very much for that. So yes, I was invited here today to talk to you about how our agency targets surveillance for agricultural pests and diseases. As mentioned, I've worked on both sides of the house. So I'll be trying to give examples as I'm going through the presentation. So I work for USDA, the Department of Agriculture, and specifically within USDA, the Animal and Plant Health Inspection Service. And one of APHIS's missions is focused on preventing the entry and spread of invasive species that can impact our agricultural and natural ecosystems. And our work is interdisciplinary. We have on staff ecologists, biologists, pathologists, epidemiologists, statisticians, economists, and so forth. And we bring our expertise together to address critical issues regarding invasive species and in protecting our national agriculture resources. The way in which we do that, because we are a federal program, we must look at the global picture. And to prevent entry, we have to look at what's coming into the country. And one of the ways in which we do that is by looking at formal trade pathways. And by formal, you can think of as commercial imports of plant and animal products and byproducts, germplasm, biologics, and also hitchhikers that might accompany those imports through ballast water, actually hitchhiking within cargo containers on the ship itself, or in wood packaging material. Given the example of wood packaging material, there's several examples of non-native wood-boring insects that have been introduced to the US through wood packaging material. So you can think of pallets and crates and so forth. And with that discovery, there was a phytosanitary standard put in place that requires treatment of wood packaging materials to prevent that from occurring. Aside from formal trade pathways, there's also informal trade. And this occurs through travel, tourism, and other transboundary movements, such as people going to visit family, going to international schools, business travel, and so forth. There's also intentional smuggling of plant and animal products using these movement pathways. And that's generally an attempt to bypass regulatory prohibitions on the import of products. So by trying to bypass these regulations, they smuggle these products into the country for black market, or to even support the exotic plant and pet trade. And of course, there's also fomites. So there can be contaminants, like pathogens that are in soil, contaminating the bottoms of your shoes or your clothing. For instance, if you visit a farm overseas that's currently experiencing an outbreak, and you can come back to the US and possibly have those contaminants on your clothing, on your shoes, on your luggage. And of course, in formal trade pathways, there can be contamination of equipment and containers and such as well. So the bottom picture here is that if people move, so do invasive species. So we are responsible for the majority of the invasive species that have arrived in the US. And there's plenty of publications that have shown that as the volume of global trade has increased, so has the number of invasive species discovered. So which of those two pathways are harder to regulate and to prevent the spread of invasive species? It's kind of a judgment call, really. They're a bit different. There are the same regulations apply to both pathways. For the formal or commercial trade pathway, there is still smuggling where individuals or parties of individuals will mismanifest items to try to smuggle a prohibited product into the country. And those smuggling activities tend to be very large volumes when they are discovered, but more often than not, kind of rare events. So they can be hard to detect. In the informal trade pathways, such as international air passengers bringing stuff on their baggage, or cruise ships, or so forth, it's a lot more common, but much smaller volumes. So when you think about the potential for introduction, it's a very different modeling approach, thinking about that potential with lots of events, but very small volumes, or sort of the single hit large volume pathway. So when talking about what are our tools of the trade, I'm going to talk about two of them today: risk assessment and surveillance. Risk assessment leverages a lot of information on the biology, ecology, epidemiology, economics, statistics, and so forth to evaluate the likelihood of some adverse event from occurring and what its impact is. So that adverse event could be a new introduction, or perhaps it's already introduced, but the adverse impact you're trying to prevent is further spread. So just sort of, the evaluation of whatever that event is. And then using that information from the risk assessment, we can design a surveillance program that collects information to support some sort of response goal, presumably to prevent that adverse effect from occurring. So risk-informed surveillance, but also vice versa, because you can collect surveillance information, which will feed back into risk assessment. So this can become a very circular sort of process. But today I'll be talking a little bit more directionally. So let's start with risk assessment and its framework. And what does that look like? Risk assessment is actually part of a larger framework called risk analysis. And this is a diagram of what that process looks like from the animal health side. There's a very similar diagram for the plant health side. It's basically composed of 4 components: identifying the hazard, doing the risk assessment, doing risk management actions, which are the mitigations and surveillance to mitigate the risk, and then communication. And although this looks like a sequential process, in reality, these things are occurring somewhat simultaneously. And as I sort of hinted at earlier, there may be an initial risk assessment to inform surveillance. And as you're conducting surveillance, you're gathering more information. And that may update what you know about the risk of the invasive species or the organism. And so that can then feed back into your surveillance. Quite often with a new invasive species, we may not know enough about the organism from the hazard ID that's done. And I'm going to talk more about that here in a minute. So sometimes we have to start with an analog species that has a very similar sort of biology and base our presumptions of what it may behave like in the environment using that analog, go out, do surveillance, collect more information, and then we can update our picture of risk. So let's start with hazard identification. This is knowing your biological agent, if it, whatever the invasive species is, if it's a pest or a pathogen. And that usually comes down to, identification in terms of, what is its taxonomy, what is its biology and behavior, its distribution in endemic areas, what countries of origin are currently affected, how does it persist in the environment, if it's an insect, what are its habitat requirements, what hosts or host systems are affected, and are there multiple hosts? Are some a more susceptible host than others? Are there wildlife reservoirs and so forth? What are the means of transmission and spread, and the types of impact involved? So you basically kind of get a sense of what this organism is, and you can basically end up answering the question, is this a potential hazard to our native system or not? And if it is, then you proceed to a risk assessment. And if it's not, then there's no need to proceed any further. It's not deemed a hazard to your system. So in the risk assessment, you then look at, okay, now that I know more about this organism, how likely is it to enter my system? Here I'll be talking about the US quite a bit. How likely is it to enter the US and spread and cause impact? So there's a lot of questions that you would end up answering, and it tends to be a very thorough research process. When we've done a lot of our risk assessments, they end up looking like 80-page dissertations, which can be quite a challenge to go through, but it's also very complete. It may be, logging information on, what is the prevalence of the hazard in the endemic areas that you have a trade relationship with. Again, how does it spread? But even more so going into the details of its pathways. And I'll be talking about pathways a little bit more specifically, but, does it move with animals or plant products? Is it transmitted through a vector, if it's a pathogen? All these sorts of questions in terms of how is it moved around? What are the methods of transmission? If it's an animal pathogen, is it, direct contact, indirect contact, aerial transmission, all these just sort of different questions that you have to be able to answer. And that gives you an idea of how it can then, spread through an introduced area. Are any of these pathways assisted by humans? You're probably going to say yes to a lot of these because, we're talking about, international trade and movement. In your particular case with, I think, a transgenic mosquito study, maybe there's a little bit less involved with human-assisted movement, but I think there could be some interesting discussion here later after the presentation. What are the susceptible hosts? Are some hosts more susceptible than others? This is where you start looking at subpopulations, where you might have differential risk across your population. Some areas or some parts of the population are more susceptible than others. If we look at the example of COVID-19, our subpopulation of humans that are age 60 and older, or with medical conditions like diabetes or so forth, are more susceptible to developing more severe forms of the disease than other parts of the population. So therefore, that subpopulation was targeted for vaccine deployment earlier than the rest of the population, and then they were monitored to see what their outcomes were like. So that's a way of targeting a subpopulation. So knowing what the susceptibility of your population is. And there's a lot more questions that you would ask, kind of going through and doing your risk assessment. I don't want to spend too much time on each of these slides. So I've been talking quite a bit about pathways, and if you're not familiar with the term, a pathway is any means that allows the entry and spread of a pest or a pathogen. And rather than just saying pest or pathogen all the time, I'll just use the term hazard. That's the term that we use because it follows the international guidelines for risk analysis. So I'll just be saying hazard for the rest of the presentation. A pathway is also composed of the mechanisms and processes by which that hazard is moved. And that can be moved intentionally or unintentionally into a new area of concern. So how can we think about breaking down a pathway in terms of what does its risk look like? One way to do that is to use scenario diagrams or event diagrams. And this is an example that I've used several times in my other presentations on pathways analysis, where we have a scenario of pork products that are potentially contaminated with a swine-specific pathogen. And those pork products are entering the US through cargo imports or air passenger baggage or some other sort of pathway. And we want to know what the risk of that is. So we might look at all the processes and mechanisms that are involved in that pathway, starting with the country of origin. And is that country that we have a movement or a trade relationship with, is that country being currently affected by the pathogen of concern? And then you start looking at individual herds that are being raised for slaughter to create pork products. So what's the likelihood that that herd is currently infected by this pathogen? If we were doing this as a quantitative approach, we could use the prevalence of the disease in that country of origin to assign that probability that the swine herd is infected. But for now, we're just kind of using this as a visual example of how to think about pathways. So let's say that there's some probability that the swine herd is infected, but not showing signs yet, and the farmer is ready to send his herd of pigs off to slaughter. Well, before he does that, does he even detect if there's anything wrong with the pig? Is it showing any signs of sickness, any clinical symptoms? And if not, or if it's too early for him to really, pick up on it, and it's not detected, then we're kind of following that line of the pathogen, which is this sort of little red icon that I've given it. So if he does detect it, then it's probably reported to local authorities, and that infected pig is mitigated, which is why I have a little X over it. It's probably a culling or some other sort of mitigation to mitigate that risk. But if it's not detected, the pig goes on to slaughter. If it is sent to slaughter, it can produce pork products or where the meat is infected, is contaminated by the pathogen and potentially infectious. It may or may not be infectious, but it can at least be contaminated with the pathogen. If the pig is not sent to slaughter, of course, you can have local disease spread. And at the manufacturing plant, there can be lots of processes that we could potentially look at that would tell us something about the risk involved for whether that pork product will come out being contaminated with the pathogen. In terms of handling and sanitary conditions and biocontrol, are the products thoroughly cooked, and does it reach the right temperature to inactivate the virus? For something like African swine fever, which is something that's really big in the news with the US right now, not that it's in the US, but it has reached the Caribbean, and so the US is on high alert for African swine fever. I'll be very specific there. African swine fever can persist in meat for months at a time, depending on how the meat was prepared and stored. So unless it is canned under high pressure and high heat conditions, there's some probability that the meat's going to be infected. And so if it's not thoroughly cooked and the virus is not inactivated in some way, it continues to be potentially infectious. There may be some quality control at the end of the manufacturing process where inspectors are looking at the products. Does it get, do they catch it or not? And if it passes QC, it's ready to be shipped out, go to store shelves, however it is exported out of the country. And then you have to look at whether the virus would actually survive transport. If you think about what's going on right now with the Port of Los Angeles and all of the cargo ships that are backed up out of the Port of Los Angeles and this additional waiting time, both in the water outside the port and sitting in cargo containers around the port itself before reaching store shelves in the US, there's a lot of additional waiting time there. And depending on the environmental conditions of the storage and the transport and so forth, would the virus survive those conditions? So there's all these different things to think about. And of course, once it reaches the port itself, we have port inspectors, and would the port inspectors actually catch that there's an illegal product entering the country? And we have such huge volumes in both the formal and informal pathways that there's no way that we can inspect everything. We can't catch it all. So there's some likelihood that it could enter the country that way. So there's all these different processes to think about in terms of visualizing your pathway. And in doing so, you can identify where the vulnerabilities and the gaps are, and where you can potentially apply surveillance and mitigations to reduce the risk at each step along the pathway, not just at the beginning, not just at the end, but all along the way. So how do we use this information in surveillance? I've talked a little bit about it with just visualizing the pathway, but how do we use it in surveillance design? So we're going to move into that now. And this information actually is a summary of what we submitted as a book chapter called Surveillance Design After Initial Detection for a book that's going to be published next year, Tactical Sciences for Biosecurity of Animal and Plant Systems. So if you're interested in following more of this information, that will be out for publication next year. So surveillance design is going to follow the type of goal for whatever your response is. And in our agency, there's several different types of goals that we can potentially have. And these goals are usually set by upper level management, some sort of goal that they want to be able to achieve. So if there's a new introduction of an invasive species in an area, we may want to know more about, well, how big is the infestation or the affected area? So we need to delimit the boundaries of that infestation. Once we've delimited, perhaps management says, well, now we want to eradicate it because there's too large of a potential impact for this invasive species in this area. We need to eradicate it. So that could be a potential response goal. During the eradication process, there might be a simultaneous goal of business continuity where there are businesses or other operations or human movements that are occurring in the affected area. And you have to do surveillance on those movements to ensure that the invasive species is not being moved around outside of the affected area. So that really comes down to pathways, right? That business continuity. Once your invasive species has been eradicated, if you're a regulatory agency like we are, you have to show proof that you have eradicated it successfully. And so this is not just, that we've gotten rid of the pest and we, collected samples and it came back zero. You have to do an ongoing survey year after year to show that it has not been reintroduced or it was not, hiding in an area that we didn't inspect. We have hunted down every last remaining organism or case that we can find. And to the best of our ability, with a certain level of certainty, this is the proof that we provide that we are hazard-free and we can resume international trade or whatever might be needed. Because with the US, if we're impacted by an invasive species, that can impact our trade relations with other countries. So hazard freedom is something that we have to provide as proof to other trade partners in order to be able to resume those international trade agreements. If we're not able to eradicate an invasive species, perhaps we're monitoring its current state. We're trying to achieve a stable health status. So if it becomes endemic and naturalizes, and I'll be giving an example of that later, we may be monitoring it to make sure that it doesn't further spread or that it doesn't have a sudden boom and require some sort of a mitigation to control that population outbreak. And for each of those response goals, there's going to be a suite of response activities associated with it, and surveillance is just one of them. And so surveillance alone does not tell your program if it's successful in achieving the goal. It just provides supporting information, because there's a lot of other information that's collected through risk assessment, through your mitigation activities, through your field investigations, through research, and your starting analog, if you start with an analog species. So there's a lot of information that feeds into your response, not just the surveillance program. And with all that activity and information, there's this sort of blowout here that I'm showing with the design of your surveillance. And this is what we're going to go into next, is how to design your surveillance. And I've hinted at earlier that this is a cycle that can be updated as you get more risk information on an ongoing basis. And we have a core set of design factors that I'll be talking about today that is part of your design scheme for your surveillance program. And there's, of course, evaluation metrics as well, but that'll be in the book chapter. I don't think I didn't have really time to fit it into today's presentation on how to evaluate your surveillance program. So let's start with the surveillance goals and the difference between your response goals versus surveillance objectives, because they're not quite the same thing. With the response goal of delimitation, the description for that response goal is placing boundaries on the extent of that infestation or that infected area. And with that are the additional activities of quarantines and movement restrictions to keep things from moving outside of that boundary area. The surveillance objective is to determine which locations, farms, premises, or habitats are within the infected area versus outside the affected. So it's a little bit different. It helps inform the boundary, but it's not exactly the same thing. Business continuity is, it provides a structure for moving products and materials without spreading the hazard. And again, there's a suite of activities that surround that, involving biosecurity measures, treatments, sanitation, cleaning, disinfecting, hold times, and other sorts of mitigation actions. The surveillance objective for that is to provide evidence that the material being moved has a reduced probability of spreading the hazard, while allowing continued business operations. So this kind of gives you an idea of how surveillance is actually a bit different than the response goal. It provides supporting information, but it does not tell you the entirety of whether your response is going to be successful or not when it comes down to evaluating the success of your response. So that was just sort of a taste. You can read the rest of these on the slide a bit later, because I believe this is going to be shared to you guys. So I'll just go ahead and move on in the interest of time to design factors. So once you have an idea of what your response goal is and defining the objective for your surveillance to support that goal, then you can start designing the surveillance itself. And these are just the 5 core factors that we came up with that should be present in every surveillance design. Of course, there are many, many more that you can consider, but this is a starting point for providing a framework. We try to give a generalized framework because surveillance is always going to be highly tailored to your individual response. And even if you respond to a hazard one year and respond to the exact same hazard the next year, there can be enough differences year to year that you may need to adjust your surveillance program and redefine your factors. So again, these are just really sort of generalized high level core factors to kind of start the conversation with. So scope is the scope of your surveillance, and targeting that to a manageable set of locations or populations that can potentially harbor the hazard. So who is the host? What is the susceptible host or population? So when we were talking about COVID-19, the susceptible population are humans. But there's also been other animal species that we found that can potentially harbor the hazard as well, like, cats and dogs and mink. Target, you gain efficiency in your surveillance program by focusing on a subpopulation or subset of sublocations that have differential risk from the rest of the population. So if you're looking for the potential for introduction and high impact, you're going to focus on the subpopulation that is most susceptible to that hazard. So again, the COVID-19 example, we targeted the elderly and the high-risk populations first with the vaccine and monitored the outcomes from the vaccine deployment. Timing, when the hazard or the information that you're trying to gather about the hazard is easiest to measure. So that might be when it's most prevalent in the environment due to seasonality and development of the pest, like if it's a pest, for instance. Pest life cycle development is highly tied to heat units and seasonality of the year. So you might be looking during a certain time of year for a particular life stage of the insect. You may also target your timing if it's based on movement and pathways. So for business continuity during like an outbreak or there's an infested area and there are movements occurring, perhaps you're doing surveillance just prior to a movement to make sure that there's no hazard associated with that movement before allowing it to pass. So that's a different example of timing. And you can have all sorts of different combinations in terms of life phases of the hazard itself, of the host system, seasonality, weather conditions, or just the timing of and limitations around the response itself. For tools, this is a very generic description, and I sort of apologize for it, but we could have done a whole book chapter just on tools and tool selection. But selecting the best tool or combination of tools to support the objective, and making note of the sensitivity and specificity of the tool, or if it doesn't have that, like if you're using pheromone traps, you may be looking at the effective area radius of a pheromone trap rather than its sensitivity. It gives you sort of similar information on the proportion of insects that you can draw to the trap within a particular radius. And the tool, it can be specific to the type of information that you are collecting. So again, for insects, if they're a flying insect, then we'd be waiting for the adult life stage. So that would be the timing aspect. And we might use a pheromone trap because during the flight stage, they're actively seeking mates, which is why they're flying around. So you'd be using a synthetic sex hormone to lure the insect to the trap. So again, depending on what it is, that the type of information that you're trying to collect and the timing and so forth, you'll select the best and most appropriate tool for that objective. And then it all comes down to numbers, like how many samples to collect, how frequently should I collect the data? And that comes down to, what the true value that you are measuring, what sort of precision is needed, the design prevalence. So how much tolerance do you have in terms of potentially not detecting it? So if we want really to be able to detect a pest at really low prevalence in the habitat, it's really rare. There's just a few individuals that are introduced, but you want to be able to pick up on it. You would set a very low design prevalence to be able to pick up those very few individuals that are out in the environment. And then, so that all that information about the value and precision, design prevalence, those are all things that are very easily discoverable in the literature with regard to sample size calculation. There's lots of information on sample size calculation, so I'm not going to dive into that so much. But something I do want to mention is that something that can sometimes be overlooked is how fast the information changes. How often do you need to sample your information to be able to understand what's going on? If you're trying to measure, let's say, a fast spreading pathogen in an environment, And that spread, I mean, you're getting new cases weekly. You better be sampling very frequently to be able to capture that information so you can keep up with the change. Otherwise, you know, if you're doing something like delimitation and you're not sampling frequently enough, you can have new cases pop up outside of your delimited area. And then you're always playing a game of catch up, of expanding your borders. Oh, there's a new case, expand your border again. So be aware of the biology and its, you know, transmission and you know, how fast does it move and spread? And is it necessary to have a very frequent design, or is something that, you know, once or twice a season is enough? And I actually have an example of we sort of set out traps for a particular insect and we left it, and that's all we needed to do for the season. So I'm going to talk now about the interaction between the design factors and the response goal. So depending on the response goal, some design factors will be much more critical than others to really think about carefully when designing surveillance. So for delimitation, it's really important to think about the target and targeting the surveillance along pathways. So in this case, these will be like sublocations, not necessarily subpopulations, although it could be. But when you're talking about trying to bound an infected area, you also want to look at how that hazard can potentially be moved around within the infected area. So if there are human-assisted pathways, you're going to want to extend your delimitation boundaries out far enough to capture those pathways and then sample and target surveillance along those pathways, just to make sure that there's that you're basically, you know, kind of finding the sort of true extent of your infected area, and have a tool that's sensitive enough to actually detect the hazard if it's there. I've talked a little bit about business continuity already. The scope is a single site, typically that business that's doing the movement, and the timing is just before the product moves. And typically you need very high sensitivity because you're trying to ensure that the product being moved does not have the hazard associated with it. So you need probably an even higher sensitivity than your delimitation program. For eradication, this is very different from the delimitation example. So once you've delimited an area and you know where your infested area is, there are eradication activities that are occurring to remove those cases or infestation from the area. But now you need to make sure that there aren't any other cases that haven't been detected yet. So now we're going to change our scope and cast a wide net and sample the rest of the potential locations or parts of the population that haven't been detected or that haven't really been surveyed yet, to make sure that we're finding all of the remaining cases. Because you can't just, I mean, yeah, you can have a local eradication, but if you're not casting a wide net to make sure that you're finding everything, you can have an outbreak pop up somewhere else. So eradication is really about finding all of those remaining detections, all those remaining cases, so you can apply mitigations to those. And then with hazard freedom, once we've achieved eradication, we're again doing a broader population sample with representative sampling, and our timing, rather than being specific to a particular season or when the hazard's most prevalent, we may do more systematic sampling to make sure that our detection is not a function of poor timing. And it's usually associated with a lot of samples, a lot high volume, and the tool has to be highly specific. So rather than highly sensitive, highly specific, because we don't want false negatives, where we say we don't have the hazard, but oops, we actually do, because our tool was not appropriate for that particular surveillance. And with stable health status, whatever the characteristic is that you are targeting and sampling, it's generally representative sampling. But if prevalence is the only focus, then you can target your sampling similar to the way you do with delimitation. And again, the timing tends to be systematic, and generally tools that have a balance of sensitivity and specificity work best for stable health status. So let's go into the example that I have for you today, and this is based on a publication that I did, which actually kind of brought me to the attention of Dr. Abrakta. And this is on European gypsy moth, Lamontria dispar. So if you're from the northeastern United States, you might know this pest. Quick hazard identification. This is an invasive forest pest that feeds on over 300 species of trees and shrubs. So it is a generalist, has broad climate suitability across the US. The only areas that are not climatically suitable are the southern tip of Florida, where it's too warm in the winter for diapause, and too cold in the higher elevations of the mountains of the US to be able to complete development life cycle. It spreads naturally by ballooning. Ballooning is the process where a first instar larvae has hatched from an egg, and it releases a silken string, which is then caught by the wind, and it lifts the insect, and it basically floats on the air until it finds some host vegetation. If it doesn't find any host vegetation, then it will crawl down and crawl back up something else until it finds a host that it can feed on. And the caterpillars feed on the canopy, on the leaves of the trees. And in sufficient densities, they can defoliate entire canopies of forest, which can then reduce mass production, the quality of the timber products, alter nutrient cycling, and even surrounding native species. This insect is also spread by humans and can reach hundreds of to thousands of kilometers away from where it originated. And the way that it does that is that gypsy moth lays very sticky egg masses that can stick to anything. The sheer sides of, metal buildings, tree bark, obviously, your lawn furniture, your grills, the mud flaps on your vehicles, your RVs and your campers. And if you go driving or camping with your vehicles, you can move the egg masses that way where they hatch in a new area. Or you can, if you move, let's say you get a job offer, you can pack up all of your household goods from an area that's infested, move cross country to an uninfested area and have those egg masses hatch and release the insect there. And that does happen quite a bit. So this insect does cause quite a bit of impact, and there's a few papers on it, some of them estimating that in excess of $250 million a year. So yes, this is a hazard, and it's actually an ongoing invasion. So European gypsy moth was introduced back in 1869, I believe, in Medford, Massachusetts, by a hobbyist entomologist, let's say, who was doing experiments with the moth. I think he wanted to produce look at the moths for potential silk production for the silk industry, and it got away from him. And there were early eradication efforts, but they were not successful, and the insect spread very rapidly throughout the Northeast. And early estimates were like 20 to 30 kilometers per year was the spread rate of this insect. Congress implemented a slow the spread program that's managed by the Forest Service, which implements mitigation activities along the spread front. So that's what I'm showing in the diagram here. The sort of tannish yellowish area is the infested area, which is also under federal quarantine. And the active spread front between the two hash lines are what's managed by the Slow the Spread program. And they have active mitigations to control outbreak populations, very localized outbreak populations along the spread front that slows the spread down to more like 5 to 7 kilometers a year. My agency, APHIS, the Animal and Plant Health Inspection Service, is responsible for the rest of the US, that big, empty, white area. And let me tell you, that's a lot of area to cover. And our goal, our mission is to detect any introduced populations of gypsy moth in this uninfested area. So how do we target that early? How do we target early detection for this insect over such a huge area? So this is the work that I did when I was still with plant protection quarantine. I developed risk maps that would inform and target the surveillance to particular locations where the pest was most likely to be introduced. And the methods that I used are known as niche models or species distribution models, where you take data points of where the pest is known to occur and intersect it against maps of predictor variables, environmental predictor variables that you think have some limiting effect on the distribution of the pest. And that's what informs the niche or the habitat requirements of the niche. Rather than doing habitat variables, what I did is I applied the same methodology, but used introduction potential variables. So population density, household income, because it might have some relevance to, whether someone's able to afford recreational vehicles and all this outdoor equipment that might have, where gypsy moth egg masses might, cling to and reside. So yeah, there's a suite of predictor variables that are more associated with introduction. And you intersect the data points of known occurrence against these maps and create a data cloud that you fit some sort of regression or algorithm into that explains the variability in your data, the likelihood of introduction. And once you have a response function, you then apply it back to your predictor maps to create a final prediction risk surface or a likelihood surface so that you can then use to target your survey to higher risk locations. One of my predictors was a human-assisted spread factor. So when I talked earlier about gypsy moth egg masses and people moving their household goods from the infested area, let's say to like Seattle, Washington, they could be moving egg masses with them when packing up their household goods. So what I did is I formed an agreement with the US Postal Service to acquire address forwarding data. So when you move your household, you submit a change of address form that says, this is your old address and this is your new address. And what I did is I filtered the origin addresses, the old addresses, to the region of the US that's currently infested with gypsy moth. And then for the destinations, counted up how many people moved to that destination. Originally, we looked at the zip code level, but there was lots of zeros and ones, so to speak. We wanted some higher counts, so we aggregated to the census tract level, which ended up doing a better job of working in the model. So we ended up having destination counts. So we had origin destination movements with the volume of movement represented as counts of people moving from the infested area to some destination location. So that's how I quantified the human-assisted spread for gypsy moth and used that as one of the many predictors in the model. And I created risk maps for each year of the survey that we wanted to do. The first risk map was produced for the survey year 2014, and we regionalized the model into 2 regions. And in 2015, we updated the model again, but this time had 3 regions, which had a smoother risk surface from the spread front to the long range areas of the rest of the US, and kept that mode of model development for 2016 and onward. There was no model for survey year 2017. There were some staff availability issues that year. So once we had risk maps, we would give that to the program manager, who then on a state-by-state level would look at, how much risk, how many high risk locations were in a state, and he would allocate resources accordingly to each state. And the state could use the risk map to allocate their number of available traps to high risk locations. So let's use the design factors for the gypsy moth example. The scope was all urban and natural forests in the APHIS survey area, which is way too broad, way too many. And like I said, there's over 300 different species that are potentially affected. So the way that we targeted was by using grid locations from the risk map that have higher levels of probability, but we did sample across the spectrum as well. So we also sampled low risk locations in addition to high ones, because we wanted to make sure that we were actually fitting the model to risk adequately. So if there were any low risk sites where a gypsy moth was actually introduced, we'd gain some information from that particular site, which could feed back into the model and help adjust the model fit for next time. So within a grid location, which was a 1 kilometer square area, we would hang the target in a preferred tree species, which was generally oak, aspen, birch, or something like that, and hang the trap in the tree at breast height. So that's how the location was targeted. And the timing was during the adult life stage, when moths were flying, because the male moths are searching for females to mate. Yeah, so traps would be deployed just before the flight season and would be taken down just after flight season ends. We generally gave about a 2-week buffer before and after that estimated flight season, just to account for model uncertainty, because we're using climate data to estimate when that life stage would occur. The tool was a delta trap, a pheromone trap, which uses a synthetic sex hormone called Disparlure. And the volume was predetermined by the budget and allocated according to the risk locations generated by the risk map on a state-by-state level. And they were deployed once annually during the flight season. And the reason for that is that these traps, the lure can last months. So they last all throughout the season. And so they only need to be deployed once, and then they're taken down at the end of the flight season, and the moths are counted. They might need to be, they are checked bi-weekly just to make sure that they're not completely full or that the trap is damaged, in which case the trap will be replaced. So once the surveillance traps have been set out for that year, according to the risk map, we collect the data and we intersect it with the risk map to look at how well did it perform. 2015 ended up being an outbreak year for gypsy moth. So there were a lot of detections that ended up far away from the spread front. You can see them in South Dakota, in Wyoming, Idaho, Oregon, and Washington. Oregon and Washington in particular also had invasion of the Asian gypsy moth that came through the seaports. Which is a different subspecies, but we're going to focus on the European example here. And the risk model actually did a very good job of predicting where those infestations in the Pacific Northwest were going to occur, which you can see in the detail in the bottom left. So let's look at model performance. 2014, as I mentioned, was the first generation of the model development. It only had 2 regions, so there was an abrupt transition in risk from the spread front to the rest of the US. And we ended up seeing that when we validated the model, so you can see this abrupt bump in the receiver operating curve for 2014. So the next year we developed 3 regions for the model. So a short range, an intermediate range, and a long range model. And they had different predictors for each part of the region. And we saw an increase in model performance for the 2015 model. So we kept that same type of region, the same regions for 2016, didn't change anything else. All we did was update the data, the survey data that informed the model, and updated the pathways data, because we were getting more and more address forwarding data from the Postal Service. And we added that to the predictors. And 2016 had another increase in model performance, because we're iterating between the risk model and the survey data And that iteration cycle is helping the model to basically tune in on the information on risk, and the model gets better and better over time. Accordingly, we found that the pathway data, the address forwarding data, became more prominent as important in terms of its variable importance over time. So in the 2015 model, it was ranked 6th in importance and explained 6% of the variability. But by the 2018 model, it was ranked number 1 as the most important predictor of introduction potential. And it explained almost 36% of the variation in the model. And that was after, so you can see the number of years of accumulated data for the address forwarding data. So as we accumulated more movements of people, the predictor got better and better in the model. And the reason for that is because it represents propagule pressure, which is an ecological concept. It's the volume of introductions per time unit. And as you get more introductions per time unit, the likelihood of establishment increases. And it's very similar to a dose response model. The reason is that with introductions, you have to overcome different sources of stochasticity, mate finding issues, environmental conditions in terms of climate or weather, overcoming predation, finding a host or source food or something else. So with just a few individuals, they're more likely to die off because of these different stochastic factors that they have to overcome. However, if you introduce lots of individuals basically at the same time to the same location, then they're more likely to overcome those sources of stochasticity and actually establish. So that's why the pathways data worked as well as it did. So in summary, good surveillance design requires intimate knowledge of the biology and ecology of the hazard and its hosts, how it spreads, and being able to match the design of your surveillance to support your goal. And also just understanding what your tolerance for risk is, what your tolerance for error is in your surveillance design. And if you're using pathways data in risk models, it's important to look at it in terms of origin destination movements and quantifying the volume of movement per time unit, because that's a more mechanistic measure of propagule pressure for an invasive species. I've done models where I've tried using distance from a location where the locations are potential sources of introduction, and they do not work well in response models. The response curves are weird, they're funky, and they just don't make any logical sense. So distance from points do not work very well in risk models. I very much encourage the use and collection of origin destination movements if at all possible. And if you don't have it, try setting up surveillance to collect that data so that you do have it and see how it affects the movement of your organisms around your area. So with that, my presentation is done. And do we have any questions?
Thank you, Gericke, for that presentation. Really very interesting. And it was a fantastic primer on surveillance, at least from my point of view, because I didn't appreciate the systematic nature by which the highly systematic nature and sort of formalized way in which you approach this. And I thought that was really enlightening from my perspective, and I'm sure for a lot of people as well. There were a lot of things in there that were really interesting to me. And I guess one of the concepts I would like to maybe just, well, a couple of things. I was very interested in, maybe I'll start with this, the modeling itself. While I'm not a modeler, I would be interested in just hearing a little bit more about how it is done, even though I'm not informed about the specifics. But this sounded like it was some sort of a machine learning kind of a system whereby, you were giving it data and then having it come up with a function. Could you say a little bit more about that?
Yeah. I do tend to use a suite of machine learning models, whether they're general linearized models or multivariate regressive splines, could be random forest, whatever sort of modeling platform you like to use. Some of them are better than others, depending on the application you're looking for. If you're doing more extrapolation of risk to areas where you don't have data points, it's better to use more generalized functions like a general linearized model or a Mars model. If you have your entire study area is representatively sampled and you're not doing extrapolation, you're doing interpolation, then functions like random forest do a very good job of interpolation. They can just potentially overfit if you're trying to extrapolate. So there's some rules, some kind of rules of thumb that I like to use when it comes to machine learning methods that fit whatever sort of response curve you have through your data. I have seen some people use GIS methodologies to try to produce risk maps where they will take their predictors and overlay them together and do some sort of a weighted overlay. And whichever ones have a higher aggregate risk have, they'll use that to target their surveillance. I did some of those initially, and when I actually tried validating it with survey data, they didn't tell me anything. Mm-hmm. It can be sort of a quickie, sort of rough idea, but there's a lot of assumptions about that sort of methodology that assumes like, a linear response when you're doing your overlay methods using GIS. So I tend to use more machine learning methods to fit some sort of a response curve, because anything that's biological or ecological in nature is going to be nonlinear.
Mm-hmm. Mm-hmm. Okay, thanks. There's a question from somebody in the audience, and it was something that I had in my mind as well. And you focused on the forwarding address as a way of weighting the volume of movement, or I don't know if I said that right, but the question is, did you also consider traffic routes, cars, trucks, and planes in your model?
Yes.
And which is, of course, happening all the time, and interstate highways, major thoroughfares, big arteries like that, and things.
Yes.
Was that in the model as well? And
it was. Yeah, that was actually a year-long project. So I had traffic volume data that was collected from various departments of transportation. So those little lines that those little wires that you drive over, and they're mm-hmm. Counting traffic. I took those data points and they were directional, and basically created a custom algorithm that would interpolate the volume of traffic directionally along roadways and basically interpolate that data as best as I could, given the data that we had, because often there would be years where they would alternate their surveys. So for a state, they might survey this region for 1 year and then survey this region for another survey year. So it was sort of interesting, but yeah, I had a data set where I interpolated the volume of traffic nationally. I think we might have a publication on that as well. And you did and that was actually mapped to road networks. So in order to create a gridded surface, we ended up doing it's a GIS tool that kind of smooths out those data. It's kind of like a density surface estimator that will kind of smooth out that traffic across the surface, and just so you can sort of sample if an introduction is close to a road with a high traffic volume, not necessarily on the road itself, because it was a road network. We want to know if it's close to. So we wanted to be able to smooth out the road network over some area, over some gradient, and be able to use that as a predictor. And you can see it in the risk maps, how there's sort of a road or network-like structure to the data. So there's some hot spots that are around urban areas, and then you can see some lines that kind of radiate out to it, and that's the road traffic that it's picking up on.
Mm-hmm.
So it is picking up on the human element of movement as well. And we use traffic volume that moves west and south because that's the same direction as the direction of spread for this insect.
And in terms of response, the model, what was, you mentioned that I guess you termed it in terms of like 30 from the forwarding address data, it was accounting for 37%. Maybe I'm just not picking up on how you could parse out how well each of those components of the model were helping you, or could you?
Yeah, so there's basically this sort of algorithm when the model is running that basically subsamples the data many times and fits the model over and over again and looks at the model. It looks at the performance of each predictor with each subset of the data, and produces a metric that shows how well that variable performs on average with all those different subsets.
Yeah. I guess in looking at, and listening to this and also reading your paper originally, which got me connected to you to begin with, I guess I see an analogy here. I'm just going to sort of articulate it, and in terms of a gene drive. So hypothetical completely, but someone is going to, and I'm thinking early in the gene drive testing phase, which hasn't been done yet outside. So this is all, hypothetical. People are thinking about it. And of course, there'll be some, as I mentioned in my introductory remarks, there'll probably be, I would imagine, that there'll be some concerns about physically or geographically or somehow containing this. And of course, one might imagine an island scenario or whatever, and then with the threat of movement, unintended movement of the gene drive product off of that geographic location or whatever to some other point in time, or some other point on the map. So that's sort of how I see, this problem that you've described. And in the area that you described, that you're responsible for surveilling, is huge. And I would imagine that, again, that this same sort of a challenge will be faced by people who are interested in, keeping those gene drive products or knowing where those gene drive products are that they're testing, and not intending to move beyond the area where they're testing them, wherever that happens to be. And that they're going to have to consider, I hope they're going to have to consider how to deal with this problem. So to me, I see this scale of the problem that you've described and the challenges as being very much analogous to what I think, when I imagine some of the initial efforts to test gene drive products, where containment will be sort of the issue here, to try to keep it contained or to know where it is at least at all times. So I really, I was really very excited. Now, obviously, movement will be something that's really important, and I'm sure that'll be considered. But one of the other things that you mentioned was proof of eradication, or how do you, I'd like you to say more about that, to have confidence that something is not there, which is really seems like a tremendous challenge. And if you could just talk more about that within the sphere of invasive species. How do you come to a decision that either an eradication has been completed, or that an invasion has been averted, or that there hasn't been an introduction, and so on?
Right. So with those proofs of freedom, it's generally a larger narrative that, it does include the surveillance data, where you give a description of the surveillance program, the design, and the samples and the results of that. And we've not detected this insect over this period of time for as long as we've been doing surveillance, but also all of the mitigations that you've done, the field investigations, other lines and sources of information that provide additional proof beyond the surveillance program that shows what you're doing to contain the hazard, to ensure it doesn't move beyond. Your protocols and your biosecurity measures, everything that you bring to bear to that response or your program as proof that you are containing the hazard. So that's kind of sort of a high level description of what the proof of freedom is. With the surveillance piece, it comes down to the statistics with what degree of confidence you have that your surveillance program is showing that the population count is 0, given the samples that you've collected and the sensitivity and specificity of your tools and the design prevalence and so forth. What is that confidence interval around 0? And is it within a tolerable level that your trade partner or whoever that you're interacting with that wants you to show proof that you are containing it, that you're meeting their requirements? So it becomes sort of this interactive dialogue with proof of freedom, that whoever you are interacting with that wants that proof, these are their requirements for proof, and you design your surveillance to detect it to that level of precision and certainty.
Yeah, yeah. The other aspect of all this that made a tremendous impression on me was imagining the resources required to perform all of this surveillance that, and you're trying to do things that will help economize that and optimize that, but nevertheless, over those areas, I mean, the surveillance must be an enormous resource drain on an organization like APHIS. Is that?
Yes. It is. It's a huge challenge, and I already talked about how the increase in global trade is bringing new invasive species to us all the time, and this is just one program that we have surveillance for. There's many.
Yeah.
So yeah, it's a huge effort. It's a challenge for sure.
Yeah.